Today, we will be discussing on the classification of protozoa and the classification of viruses. So first, we'll see the general characteristics of protozoa. So these protozoa are eukaryotic unicellular protists that grow in a wide variety of moist habitats. Most of them are free-living and inhabit freshwater or marine environment. So these protozoa, they undergo two types of heterotrophic nutrition, that is holozoic and saprozoic nutrition. In holozoic nutrition, solid nutrients such as bacteria are acquired by phagocytosis, which lead to the formation of phagocytic vacuoles. Whereas in saprozoic nutrition, soluble nutrients such as amino acid and sugar cross the plasma membrane by pinocytosis or diffusion or carrier-mediated transport. Many of the protozoa possesses a capacity to develop into a resting stage that we call it as cyst. Cysts are the dormant structure marked by the presence of a valve and has a very low metabolic activity. Under this, there are two very interesting terms, that is encystation and excistation. We'll see what are these two. So, encystation is the formation of cyst from vegetative cells, whereas the transformation from cyst to vegetative cells are termed as excistation. Next, when we talk about the movement of protozoa, most of them are motile and move by one of the three types of locomotor organelles, that is pseudopodia, flagella or cilia. And for reproduction, they mostly reproduce asexually by the way of binary fission, but some also carry out sexual reproduction by conjugation. Now coming to viruses. Viruses are infectious agents whose distinctiveness resides in their simple acellular organization and pattern of reproduction. A complete virus particle is known as virion, and it consists of one or more molecules of DNA or RNA, which is enclosed in a coat of protein. Viruses exist in two phases. The first one is intracellular and the second one is the extracellular phase. So they exist as virions in the extracellular phase, and in their intracellular phase they exist primarily as replicating nucleic acids. And these viruses, they differ from other living cells in many aspects. So those includes their simple acellular organization and their absence of both DNA and RNA in the same virion, and their inability to reproduce independent of cells and carry out cell division. So next, we'll see the classification of protozoa. Protozoa is considered as a subkingdom of the kingdom Protista. Protozoa contains 7 of the 14 phyla that is found within the kingdom Protista. The 7 phylum includes phylum Sarcomastigophora, phylum Levantomorpha, phylum Apicomplexa, phylum Microspora, phylum Acetospora, next is phylum Myxozoa, and finally phylum Coeliophora. So now we'll see the phylum Sarcomastigophora. In the phylum Sarcomastigophora, there are 2 subphylum, that is subphylum Mastigophora and subphylum Sarcodina. These Mastigophora have a single type of a nucleus and possess pseudopodia. The subphylum Mastigophora contains both phytoflagellates, that means it contains chloroplast in their cell, and hence the name phytoflagellates, and the zooflagellates. So these zooflagellates do not have chlorophyll, and their mode of nutrition is either by holozoic, saprozoic or symbiotic. Some of the zooflagellates include Giardia lambia, Trypanosoma brucei, Trichomonas vaginalis, etc. So this Giardia lambia causes severe diarrhea in human, and it is transmitted through water that is contaminated with fecus. Further, Trichomonas vaginalis causes sexually transmitted diseases, and they reside in the vagina and urethra of women and in the prostate, seminal vesicles and urethra of men. Then when we come to Trypanosoma, Trypanosoma brucei rhododendens is a blood pathogen of human and animals, and they cause a disease that is known as African sleeping sickness. So next, the subphylum Sarcodina contains the amoeboid protease. These Sarcodina move by using pseudopodia and have no definite shape, and their internal structure occupy no particular position in the cell. In this subphylum, reproduction is carried out by binary fission. And some of the marine amoeba includes the foraminiferans and radiolarians. The foraminiferans are found mostly on the sea floor, while these radiolarians are found on the open sea. And also there are over 40,000 species of foraminiferans, and 90% of them are fossils. And a very striking contribution of these foraminiferans is the massive cell accumulation to geologic formation, that is during the Tertiary period, that is about 230 million years ago. They are so abundant that they form thick deposit which became uplifted over time and exposed as the great beds of limestone in Europe, Asia and Africa. Finally, Entamoeba and the Endomoeba are the two common symbiotic amoeba. So this Endomoeba blattae is commonly found in the intestine of cockroaches, while the Entamoeba histolytica is an important human parasite which produces severe amoebic dysentery. Next we'll see the phylum Labyrinthomorpha. These are the protists that have a spindle shape or spherical non-amoeboid vegetative cells. So most of the members of this phylum live in the marine habitat, and they are either saprozoic or parasitic on algae. So next we'll see the phylum Apicomplexa. Apicomplexans, also known as the sporozoans, are the spore formers, and they lack the special locomotory organelles. They are the parasites of animals, and they are distinguished by their unique arrangement of fibrils, microtubules, vacuoles and other organelles, which are collectively known as apical complex. The life cycle of apicomplexans is complex, in which certain stages occur in one host, that is mammal, and the other stage in a different host, that is mosquito. Their life cycle has both asexual and sexual phases. At some point, an asexual reproduction that is known as schizogony occurs. So what is this schizogony? Schizogony is a rapid series of mitotic events producing many small infective organisms through the formation of uninuclear buds. While sexual reproduction in epicomplexans involves the fertilization of a large female macrogametes by a small flagellated male gametes, and hence the resulting zygote turns into a thick walled cyst that is known as oocyst. Meiotic division occurs in the oocyst and produces infective haploid spores. And some of the sporozoans parasite includes the Plasmodium, Cryptosporidium, Toxoplasma, and so on. Next, we'll see the phylum Microspora. These microspora are obligate intracellular parasite and they lack this mitochondria. The infective stage of microsporum is transmitted from host to host as a resistant spores. So parasites under this phylum are of a great economic importance because of their role as a biological control agent for certain insects. So we'll take a unique example, that is Nosema locustae is an approved and registered by the United States Environmental Protection Agency for use in long-lasting control of rangeland grasshoppers. Next we'll come to phylum Acetospora. Acetospora is a very small phylum consisting only of parasitic protists, and these are characterized by spores lacking polar caps or polar filaments. One of the example is Haplosporidium. This is an acetosporum and are parasitic in cells, tissues and body cavities of mollusks. Next is phylum Myxozoa. Myxozoans are parasites of freshwater and marine fishes. They also possess a resistant spore with 1 to 6 coiled polar filaments. Myxosoma cerebralis is a myxozoan that causes tumbling diseases in trouts and salmon. They infect the nervous system and auditory organ, and the fish lost their sense of balance and tumble erratically, and hence the name tumbling disease is given. Now we'll see the phylum Coeliophora. The phylum Coeliophora is the largest from among the 7 protozoan phyla. So these are unicellular heterotrophic protists and use cilia as one of the locomotory organelle. And the most striking feature of the ciliate is their ability to capture many particles in the very short time by the action of cilia around the buccal cavity. When they captured food particles, food first enters the cytosome and passes into the phagocytic vacuoles, and further it is fused with the lysosome, and after it has been detached from the cytosome. The vacuoles content are digested further by the digestive enzyme released by the lysosome. After the digested material has been absorbed into the cytoplasm, the vacuole fuses with cytoproct and empties its waste material to the outside. In this way, the process completes. Further, reproduction in ciliates is carried out asexually by transverse binary fission and sexually by conjugation. Ciliates which live as commensals include Entodymium in the rumen of cattle and Nyctoteres in the colon of frogs. Some of the ciliates are strict parasites. One of the example is Balantidium coli resides in the intestine of mammals, including human, and they cause severe dysentery. Next, we'll come to the classification of viruses. The most commonly used classification of viruses is the Baltimore classification. This was developed by an American biologist, David Baltimore. Baltimore classification scheme grouped viruses into 7 classes based on their mode of replication and genome type. So according to this, viruses are placed in one of the 7 following groups. The first one is Group 1, which consists of double-stranded DNA virus. Group 2, it consists of single-stranded DNA virus. In the Group 3, it contains double-stranded RNA viruses. Group 4 contains positivesense single-stranded RNA viruses. Group 5 contains negativesense single-stranded RNA viruses. Group 6 contains reverse transcribing single-stranded RNA viruses. And finally, Group 7 contains reverse transcribing circular double-stranded DNA viruses. So now we'll see first the Group 1, that is the double-stranded DNA viruses. The viruses under this group must enter the host nucleus to replicate. In addition, they also require host cell polymerase to replicate, and hence they highly depend on cell cycle. These viruses may induce the cell to undergo cell division. This will lead to the transformation of cells and ultimately develop cancer. Some of the examples are herpes viridae, the adenoviridae, and pepoviridae. Next, coming to Group 2. Group 2 consists of the single-stranded DNA viruses. Viruses under this group have circular genome, and they mostly replicate within the host nucleus, and that is by rolling circle mechanism, and forms a double-stranded intermediate in the process. Here, rolling circle replication is nothing but it's an unidirectional replication that rapidly synthesizes multiple copies of circular molecules of DNA or RNA. Aneloviridae, Sarcoviridae, and Parvoviridae, which infects the vertebrates, the Geminiviridae, and Nanoviridae, which infects plants, and microviridae that infects prokaryotes are some of the viruses that comes under this group. Next is the Group 3, the double-stranded RNA viruses. This group of viruses possess double-stranded RNA genome. Replication in this group is monocistronic and include individual segmented genome. That is, each of the genes codes for only one protein, unlike other viruses. And families that comes under this group includes the Reoviridae and the Birnaviridae. Next, we'll see the Group 4 viruses, that is positivesense single-stranded RNA viruses. Group 4 viruses, they reproduce in the cell cytoplasm, and also the viruses in this group are further divided into 2 groups. In the first group, it includes the viruses with polycistronic mRNA, where the genome RNA forms the mRNA and translated into a polyprotein product, which is further cleaved to form the mature protein. And in another group, viruses with complex transcription, for which subgenomic mRNAs, ribosomal frameshift, and the proteolytic processing of polyproteins may be used. And Astroviridae, Calciviridae, Coronaviridae, Flaviviridae, Picornaviridae, Arteriviridae, and Togaviridae are the viruses that comes under this group. Now coming to Group 5, these are the negativesense single-stranded RNA viruses. In this, The genes of this group of viruses cannot be directly accessed by host ribosome to immediately form proteins. So, they must be transcribed by viral polymerase into a readable form, which is the reciprocal of positive-sense RNA. Arenaviridae, Orthomyxoviridae, Paramyxoviridae, Bunyaviridae, Phylobiridae and Rhabdoviridae are the families that comes under this group. Next, coming to group 6 viruses, these include the reverse transcribing single-stranded RNA virus. These viruses use the enzyme reverse transcriptase to convert the positive sense RNA into DNA. They use DNA to create the template, which is then spliced into the host genome using integrase. Here, integrase is an enzyme that enables its genetic material to be integrated into the DNA. Then further, replication commences with the help of host cell polymerase. The retroviruses are the example of this group of viruses. Now we'll come to group 7. Group 7 are the reverse transcribing circular double-stranded DNA virus. Under this, viruses have a double-stranded gapped genome, and that is subsequently filled in to form a covalently closed circle that serves as a template for the production of viral mRNA. So, Hepatnaviridae is one of the examples of this group of viruses. So, that's all about the classification of protozoa and the classification of viruses, and I'll conclude by stating that virus classification is the subject of ongoing debate and proposal. This is due to the pseudo-living nature of viruses, which are not yet definitively living or non-living. Hence, they do not fit neatly into the established biological classification system.